Cellular respiration generates 30 ATP from one glucose. But how does that happen? So we start with our glucose. And first, hexokinase catalyzes the phosphorylation of glucose. And it uses 1 ATP in this reaction. So we're down 1, and that forms our glucose 6-phosphate. Next, it's isomerized to fructose 6-phosphate with phosphoglucose isomerase. And then that fructose 6-phosphate takes an ATP and uses phosphofructokinase to form fructose 1,6-biphosphate. So note that now from this one glucose we're down 2 ATP. Next, andylase comes in and it actually forms 2 different 3-carbon structures. So andylase catalyzes the cleavage of the fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, or GAP and DHAP. But we can also use triose phosphate isomerase, or TPI, right here to convert our DHAP into GAP. So now we have 2 GAPs going through the system, and the rest of this stage 2, we have to remember that there are 2 molecules going through it. So the next part that happens is GAP is oxidized by NADH, or NAD plus, and H2O, and does a dehydration reaction forming 1,3-biphosphoglycerate. After that, our 1,3-biphosphoglycerate phosphorylates this ADP into ATP with phosphoglycerate kinase. So that gives us another net ATP. So now we've lost an ATP, gained an ATP times 2, and then next, and that leaves us with 3-phosphoglycerate, and then phosphoglycerate mutase moves this phosphate group up to here and forms 2-phosphoglycerate, and then enolase removes this water, the oxygen and 2 hydrogens from there, and then leaving us with phosphoenol pyruvate, and pyruvate kinase then uses that phosphoenol pyruvate, removes that phosphate and puts it onto ADP, onto ADP, forming ATP and our pyruvate atom. So if we go back, lose an ATP, gain 2, gain 2. This leaves us with a running total of 2 ATP, which is why they say that the anaerobic process of glycolysis forms 2 ATP. That's where that comes from. So next we have our the oxidation of this pyruvate, which is broken down into 3 steps. We can see here the first step is the decarboxylation, and it combines with the carbanion form of TPP, forming this hydroxyethyl TPP and CO2. That's the decarboxylation step. And so next, this is ionized, forming our hydroxyethyl TPP. Notice this negative charge here. So that can react with lipoamide, and that regenerates our carbanion form of TPP here and leaves us with acetyl lipoamide. And this actually makes our energy-rich thioester bond. Next, coenzyme A reacts with the acetyl lipoamide. Oh, and both of these reactions are done with pyruvate dehydrogenase as the enzyme catalyst. So next, our coenzyme A reacts with the acetyl lipoamide, forming our acetyl coenzyme A, which was the goal of this reaction, but not for what we're looking at, and dihydrolipoamide. And this is done with dihydrolipoyl transacetylase. And Once this is done, we need to transform our dihydrolipoamide back into lipoamide, and that's done with a few oxidation steps where it's oxidized by FAD, and that leaves us with FADH2, which then reacts with, gives those protons to NAD plus, forming our FAD and NADH. So notice we're forming an NADH from this step, but this FAD is conserved after it gives its hydrogens to NAD+. So, from that reaction, we gained 2 NADH. So now we're up to ATP and up to NADH from the glycolysis part alone. So next we go on to the aerobic part, or the citric acid cycle, which is summarized right here. So our oxaloacetate reacts with our acetyl coenzyme A here that we got from our glycolysis and uses citrate synthase to make our citrate molecule here. And then aconitase makes our isocitrate, which basically just swaps where the CH2 and the COO are. And then our isocitrate, finally we have our NAD plus coming in with isocitrate dehydrogenase, oxidizing it to alpha ketoglutarate, and that forms an NADH and a CO2. And then our alpha ketoglutarate again oxidizes to succinyl coenzyme A with alpha ketoglutarate dehydrogenase complex, and again we have NAD plus coming in and NADH leaving and a CO2. So far we have 2 NADHs made here. Now our succinyl coenzyme A reacts to form succinate with succinyl coenzyme synthetase, and that phosphorylates our ADP into ATP. So we're gaining an ATP here, and our succinate is oxidized into fumarate and also makes a FADH2, and that's using succinate dehydrogenase. And so now we're up in FADH2. Now we're up in NADH, up in NADH, up in ATP, and up in FADH2. So we have our fumarate, and reacts with the water from fumarase and makes our malate. And then our malate, with the help of malate dehydrogenase, reforms this oxaloacetate, restarting our cycle, and that also makes another NADH. So far we have 1 NADH being made, 1 NADH being made, so that's 2, an ATP, an FADH2, and then another NADH. That makes 3 NADHs, 1 FAD, and 1 ATP. But remember, this is from our single glucose, so we had 2 going into this cycle, so we multiply everything by 2, so we're getting 2 ATP, 6 NADHs, 1, 2, 3 times 2, 6, and 2 FADH2s. So our last step is our electron transport chain, which produces the most ATP, and our chemiosmosis. So here we have our electron transport chain, and I don't think we should go into too much detail other than in complex 1, our NADH is turned into NAD plus with NADH reductase. So all of those NADHs that we produced in the previous cycles are being used up. And also our FAD, in this picture it doesn't show the complex 2, which is our succinate dehydrogenase complex, but that forms FAD from FADH2. And then eventually this makes a gradient where there are more hydrogens concentrated out here than there are in the mitochondrial matrix. And then this goes through our ATP synthase, which has quite a complex mechanism. But basically, these hydrogens bind to the A subunit on this, which in turn turns the C subunit and this gamma pole, I believe it, and then that allows these alpha and beta units to generate ATP from ADP and phosphate using the binding change mechanism that we read about in our book. So, for our new grand total, we had 2 ATP formed from our glycolysis and 2 NADH formed from the oxidation of our pyruvate. Next, we had 2 ATP formed from our citric acid cycle and 6 NADH and 2 FAD2H. This gives us a total of 4 ATP, 10 NADHs, and 2 FADH2. So this is just a little chart to keep track of our net gains and losses throughout our reactions. So in glycolysis, remember we formed 2 net ATPs and 2 NADHs, but because the NADH in glycolysis can't directly transfer its electrons and instead has to use a shuttle system, the actual ATP gained by that per NADH is about 1.5, as opposed to in later parts where it's 2.5, like when we did our pyruvate to our 2-acetyl acid coenzyme A's, that generated the 2 NADHs, but those can generate 2.5 ATP each, so that gives us another 5. And then in our last steps, we generated 2, it could be GTP depending on the cell, but we generated our 2 ATP, our 6 NADH, and 2 FADH2. Remember, because it's 2 of these going through instead of 1 glucose from our 1 glucose. So our 6 NADHs times 2.5 is 15, plus our 2 FADHs times 1.5, which is 3. That adds up to a total of 20, giving us, plus our other 10,